Cells are very complex assemble of molecules, and just a very tiny part of the cells is the lipid membranes, which serves as the gate between the environment and the cellular interior. It's a very thin fluid layer that covers the surface of the cell, and the lipid membrane plays a very important role in protecting the cell from the environment, deciding or selecting things that go in the cell or go out. Lipids are often represented as a structure that have a round head group and two little legs. So part of the molecule is hydrophilic, water-loving part, and another part is hydrophobic, which doesn't like water. So one way to study the membrane of the cell is to remove the complexity of the cell by preparing a much simpler artificial systems made just by the lipid molecules.
So we create a model of our system, often using some kind of approximations here or there, and hopefully we can justify these approximations and show that they're actually they still lead to a good model of the system. And then from that we can extract the behavior of our system and aim to compare that to what we see in experiments. What we do is we first sort of discretize our membrane, so we split it up into lots and lots of really tiny pieces, and then we can relate how these tiny little patches of membrane interact with each other. So can the membrane stretch, and is there some sort of bending that you can describe by this sort of network, this mesh that describes your surface?
So I find membrane very fascinating, simply because they're more made of all sorts of objects that have no reason to be together except that they hate their surrounding. Because of this, it means that the interaction that makes it hold together are very, very weak and can change and can move and can evolve. So it's like a passive gathering of people who are just brought together by the current or something like that. And so it means that you can have a whole host of new science that start emerging there, because the truth that was applying in one area or the other is not kind of there. And you can see sometimes just a compromise, and sometimes you can hear an emergence of a completely new law that come there. And it is really exciting. So the same happens in many ways with membrane, because they come together precisely not because of wanting to be one medium or the other, but brought together by staying at an interface. That's how they like to be, at the interface always.
Lipid membranes are fragile structures, and if you stretch them by more than 2%, they form holes, which we call pores. Now, in cells, formation of pores is not a favorable process, because it can kill easily the cell. Therefore, cells have invented various mechanisms in order to protect their surface area by adding lipids or exploiting proteins that stabilize the membranes. We also use or exploit the pores in artificial systems, whereby we create these vesicles which can open pores under certain conditions and deliver their content, and then close the pores when these conditions are removed.
So ultimately, what really get me interested is interfaces, and that's how I got into membrane. They're just one type of interface. When you have two very different media, and somehow they have to meet up and find a boundary between them, you need to find this kind of strange environment when kind of the law of both worlds apply to some extent.
Out of this research, one of the questions, one of the experiments that we want to try now is to see whether we can use these biohybrid structures to deliver bacteria to places in the body where the microbiome has been compromised.
So getting inspiration in research can be both exhilarating and challenging at the same time. You can align whatever you want. You can align the stars if you want to make sure that all the result goes your way and try to do what you're planning to do. The reality is that 9 out of 10, it will not work the way you want. And you're going to get frustrated because the results are not at all what you're expecting or not in the way you were expecting them. And then that's when chance happens usually. And if you look at it with an open mind, you actually see that there is something else there, something you had not anticipated. And that's, to me, the whole point of it. That's the whole point of this creativity, is to be able to have this openness and this discerning eye to be able to pick up and say, hang on a minute, there is something exciting there.
Scientific research is a very creative process because, especially experimental research, you don't just reach out and take experimental technique that you can just use and measure something. It's a constant interaction and interplay with this technique where you have to make it play with you, go along with you, and collaborate with you. And this is also how you collaborate with people, how you collaborate with the techniques, how you collaborate also with the living cells you're investigating. One foot is in the known and the other one protrudes in the unknown. And then you gather the courage to really step into the unknown and trust the research where it brings you.